And so what I did to make LOCKR was I took a trimer, made it into a single chain, so now it's just one single scaffold. The key binds to that same interface, right? So that the key unlocks the latch from the cage. It's almost like a biological light switch. When it's off, it's whatever function it's tied to is inactive. But then when you switch it on at the activator, you're activating or turning on whatever function it's programmed to turn on. Control is really the main selling point of LOCKR in these kind of applications. Because it's so modular, because it's so tunable, we can really tightly control the behavior of those cells, whereas previous strategies for doing so have been insufficient so far. Our collaborators at UCSF have a kind of a sibling paper alongside this one where they've actually implemented feedback control upon naturally occurring proteins in the cell using Degron LOCKR. And so that's a new capability of synthetic biology that without LOCKR is probably would have been a lot further out than now. Feedback control is just one way of how we can engineer behaviors into cells. The potential of LOCKR is more than what is strictly published in this paper. I like to think of it as we've created a kind of a platform technology that people can build upon. And kind of like the transistor allowed kind of revolutions in electrical engineering, we're hoping that this kind of LOCKR switchable technology can kind of create similar revolutions in synthetic biology.
So David's lab has really pioneered this field of de novo protein design, being able to design proteins completely from scratch. But a really outstanding challenge has been how do we design things that are functional and that have moving dynamic parts, which is LOCKR is really one of the first examples of that. So one of the most promising new things in medicine is cellular-based therapies, and that's especially true in cancer for CAR T-cell therapies, where you can take patients' T-cells, re-engineer them, and put them back in to be like heat-seeking missiles for the tumors. But to get CAR T-cells to work for many different types of cancer, we have to engineer all kinds of new functions into them to make them really effective at killing the cancer. And LOCKR is one way that we can control these functions in the cells very precisely and engineer all these new things into the cell. I really believe that cellular-based therapies, cells as medicine, is going to be a really big part of how we treat people in the future and open up a lot of new promising therapies. And I really think that LOCKR and de novo protein design can be a big part of that. This opens up an entirely new way to engineer cells, and we're really excited about that.